Hi, good morning, and welcome to the ZP vlog and podcast. But I should actually say this is the ZP Developers Zone. Now we have come online an hour early. It does say 8 a.m. UK time there, and I have come on approximately 1 hour 7 minutes early, and I apologize for that. Now the reason we're doing this is because today we're at a conference. You can appreciate you've got to kind of have breakfast, get in the cab, and all sorts of stuff. So apologize that we're coming early, but I will basically just jump into it and start going through it. So every week we put up this slide. This slide is really just to represent that we have our ZP Academy. So if you're interested in sort of free material from ZP in terms of educational material, there's that. We do this webinar once a week at 8 a.m. London time, which is really just answering questions. We do collaborations, so mostly with a lot of UK and Norwegian universities at the moment. We do offer jobs. In fact, there's quite a lot going on at USN at the moment, the University of South East Norway, where we do a lot of recruitment from. And we do have our ZP Developer Zone website. And finally, we do have our workshops. There is a workshop next week, and I shall really cover that essentially in the next slide. So we do workshops, one in the UK the first week of the month, and one in Norway in the second week of the month. So we did have, sorry, I should, it's the other way around. We have a workshop in Norway first week of the month and in the UK in the second week of the month. So we do have one next week. Now, if you see a link, I've already put the links underneath the video, and I hope those are essentially, if I can say it this way, useful to people. I will go ahead a little bit quicker now and just say that every week the purpose of this workshop, or this webinar rather, is to bring questions that have come in during the week and answer them during this particular webinar. So questions that we've had this week are, somebody's very interested in routes to market. These are really commercialization questions, but they're very relevant. A lot of our questions can be quite technical, and we do have a lot of technical information out there, but at ZP, the kind of whole raison d'etre is actually to get people to market. Then we've also got some questions about sensors for bioreactors. This is something we've covered in the past, but I will cover it again. Also, somebody's asking about setting up a new lab and starting with biosensors, so I will cover that. Also measuring cells, both red and white blood cells in every human samples, technologies, and what's current practice. And then also, finally, microneedles for interstitial fluids analysis. We will also cover that as well. I just want to make sure that everything's going okay. Yeah, cool. All right, so if I go forward a bit. So the questions that have come in, what steps would be needed to further develop something into an ISO certified IVD sensor? To get something approved in Europe or in the US is quite a journey. It could be, with ZP, it could be a 2 to 3 year journey. And I think these kind of program costs can be something like 1.2 million to about 1.2 million to something like 2 million. They can be, just expensive basically, and maybe even up to 3 million. I will break that down a little bit and show where I'm actually getting these numbers from. I was told once upon a time by a gentleman who developed a glucose strip, and his kind of comment was, well, I spent 1 million developing my IP. I spent 10 million really developing my glucose strip and sort of getting it manufacturable, and I spent 100 million actually winning market share. And those numbers, sort of, 1 million in his first raise, 10 million in his second raise, and 100 million in his third raise, they're not ridiculous. You'll see it from from some of my data today. But I will illustrate that a little bit deeper as well. How long will it take? Now, we have got people from sort of 0 to CE marking in generally 2 to 3 years. People come to us and say, I want to do it in 1 year, but 1 year is just really too short. You don't know, and you don't always know the all of the risks up front. And so when you discover a risk, if you don't have enough time in the budget, if we find an unknown risk and then we have to solve it, so we really do need, sort of 2 to 3 years. We've done things quicker. We've done things in 1 year, but it's never too super satisfying. Let's put it that way. At what cost? I have kind of been a bit reluctant on this because, but I would say that we do have clients on the market, and they've easily spent 2 to 3 million just on us. But then they've also had to raise money to do their own clinical trials and support their own team as well. So that number earlier on, when I said, look, the first phase is often 1 million, the second phase is often 10 million, and the third phase is often 100 million. Those are not ridiculous numbers. If somebody wanted a sort of a first for research only kind of product, we could probably do it in 3 to 6 months. In fact, we have done that kind of thing. But the first deliverable, for example, we gave on one program was about a 6-month effort. It wasn't very satisfactory. The nurses were pipetting, and asking nurses to pipette is not a great solution. But to be fair, they did do it, and the clinical trial went okay. So we can kind of do this kind of work in, I would suggest, 3 to 6 months. Now that means people are staying very close to the platforms we already have in place, and not trying to do something that's very far from what we've already got in place. What's the eventual production cost of a sensor? Eventual production cost of a sensor in low volume. So first of all, it's an answer I've given in the past. It's an answer I can always stick to. In high volume, a glucose strip, and there'll be examples of kind of capillary fill sensors in this presentation today. A glucose strip at volume, they can make those for 2 to 5 cents each, and that's obviously there's what we call a substrate. They've put some electrodes onto it. They've deposited their enzyme on it. They built a little microfluidic capillary channel on it, and they can then, as I say, sell that for 2 to 5 cents. But that's at volume, and that's after 40 odd years. It's probably reasonable to say that if you're much lower volume and you're much earlier on in your commercialization, sometimes these kind of sensors can cost, like, especially in an R&D phase, can be kind of 80 euros each, just because it's so small volume and there's a lot of manual effort involved. 80 euros is not is not super surprising. I think we can get most things down to about 10 euros once we've done quite a lot of engineering towards that. If you really want to know where the floor is on this, it's that 2 to 5 cents, but it really takes a lot of effort to get to there. Now, this person was asking a lot about IVD, in vitro diagnostics. They also had the sort of same questions for continuous glucose monitoring. At ZP, we do actually have experience, obviously, in IVD and continuous monitoring. A lot of work at ZP is in IVD, and we also do a lot of CGM, continuous glucose monitoring. But I think we have more projects that have been IVD. So when I give these numbers about millions and months, etc., it's actually because we've got a lot of experience in it. On the CGM, we have less data, or continuous, we have less data points. So I'm only going to give one data point on CGM. I'm going to give the data point of BioLink in a bit. So I do have a slide on this as well. Now, I suppose what I would say is what ZP does do is we actually do this kind of planning for people. So people come in to us and say, look, this is what I want to achieve. But we sit down and we can do something like 100 hours worth of planning on this. You have to get a lot of engineers in the room. You have to kind of look at the requirement specification, and then you try to sort of plan out in a Gantt charts, how long this is going to take. So we do that, but it's no trivial effort to really plan this stuff out. So at the moment, I'm just giving opinions, but I will give a particular example of the cost and time it's taken a particular company to do a CGM, and hopefully that will illustrate the point. just for your interest, I've been kind of throwing these numbers of 1 and 10 and 100 million out. I've got a few sources of that. Some that have been told to me. There's also some data here. So iStat is an IVD device. Biosite, these are all either sort of handheld or desktop type instruments. Several of them are electrochemical, and it just gives you a sense of the funding that they have had to achieve in order to get to the market. So the average cost for these devices, now the data is spread out from something like 1984 to 2005, was 38 million. So, if you're looking for kind of case studies or examples of what it has cost people, iStat, Cepheid, Abaxis, Biosite, Epocal, Sphere, Claros, Tierlab. Now, Tierlab was an exceptionally simple device. Well, they did it for less than 10 million, but if I remove Tierlab, because I think it's exceptionally simple, whoops-a-daisy, then it's 42 million on average. So I bring this up because I'm giving kind of anecdotal opinions earlier on, but I also want to say that, this study was actually done after all those, I gathered all those opinions, but the study kind of backs up what I'm saying. If we adjust these pricing, so some of that pricing, for example, iStat, that was developed in 1984. Obviously, we've had a lot of inflation since 1984. So there's something called the CPI, the Consumer Price Index. I've adjusted all the prices and said, right, if we were doing this today, what would it cost? And the cost today would be, if we did it 2021, all those numbers would come to 74.7 million. If you removed Tierlab, whoops-a-daisy, it's 83 million. So I think really in line with what I've said, that these people, in order to get regulatory approval, so a lot of these are US examples, but honestly, it's not an order of magnitude different if you do it in Europe as well. Now, I think that ZP is actually really efficient. Actually, I'll give an illustration of why I think that's why we at ZP are super efficient. If we look at one of the charts from earlier on, the average time it took people to develop their products was 7 years. And in fact, I'm going to talk about Biosite in a minute, and they've been doing this for 7 years to this point. Now, iStat and Epocal were the same company, same founder. The first time he did it for approximately in 1983, and then 2001. When I say he did it again, what I mean is he made a fairly similar product, very separated in time, but I would argue that he took all the learning that he made in his first product, and I suggest that he took a lot of learning from the first product and put it to the second product. But anyway, in doing that, he's reduced both his time and his costs, and he's reduced the time by 1/3. So he went down from 9 years to 6 years. I still think that could be compressed some more. And his costs, he dropped them by sort of 50% in really gaining that experience from the first time round. Now, in continuous monitoring, I'm going to use a real world example. By the way, if any links ever jump up, all links I've already put them underneath the YouTube video this morning, so links are already up. This is I realize that this actually supports a lot of the comments I just made. So BioLink have been running for approximately 7 years. About 18 months ago, they raised 100 million US dollars. I don't know firsthand, but I suspect that they would want to raise more money at this point. And they're doing the CGM, continuous glucose monitoring. Now, the 100 million is not to win market share so far. This is actually developed technology. And so I just want to give you a sense that if your interests are in microneedles, which is I would describe as minimally invasive interstitial fluid monitoring, then people have so far invested over 100 million in BioLink, and they've been doing this for about 7 years. So it gives you a good sense of effort and cost. Now, we do actually have something called ZP Accelerator, where we do actually have a lot of this technology stack in place already. I think what ZP does is some of us have been involved in continuous on-skin monitoring for about 15 years. So what really happens then with ZP is people actually get that almost that 15-year head start, because we've been doing this for quite some time. But we do have an entire stack of technology called the ZP Accelerator. This will appear in, I want to say, several times today, and I will bring that link and that data back up in a minute. Sensors for bioreactors. I am changing gears here. I'm changing questions here. But we do get a lot of people asking, can I put ZP sensors into bioreactors? The answer is you can. I would describe that we're actually in the wrong format. We're in a great format for us, but this is basically trying to do continuous monitoring now, but in a bioreactor system. So there needs to be a certain amount of engineering done in order to make our sensors fit and operate continuously in a biosensor. We are very interested because monitoring of any industrial process is really part of Industry 4.0. Bioreactors, cell cultures, these are really important parts of sort of modern industry. And we do have this technology stack where we're able to put sensors into solutions, send the data to apps, for example, send the data to the cloud, and we can analyze it there. The reason I'm kind of happy to describe the entire technology stack is I am super concerned at any time about drift, loss of activity, and then details of things risks that we didn't perceive. So we're quite happy to be involved in the idea of putting sensors into bioreactors, but I think we need to do these things as part of projects. Otherwise, people can buy our sensors and do it for themselves. It's just you carry all the risks, and you have to kind of answer all the questions, let's say, yourself. I did have a question this week about people wanting kit for a new lab. So if you were starting out with a new lab, and I'm just highlighting here a video that we put out this week about potentiostats. So I will put that link underneath the video for that. Hopefully I have, but I think I may have missed that actually, but I will add it. So if you start out with a new lab, we have the Anapot. This is I describe as a little DC instrument. What I mean by that is it doesn't have AC capability, so it can't do EIS, electrochemical impedance spectroscopy. If people are interested in electrochemical impedance spectroscopy, then we do have an EIS version. I would suggest that if you're in a research phase, it's good to have these EIS instruments. There's no need to leave EIS out of your capabilities. Electrochemical impedance spectroscopy is a very useful technique. If people are very interested in sort of developing products and OEM solutions, we actually do have those kind of electronics as well, and I put a couple of links underneath that as well. If somebody's just trying to fit out a lab, we do have a really nice range of carbon, platinum, and gold electrodes these days, and I mean basically the pricing is ridiculously low. But we do that because we want, essentially, I think the whole raison d'etre is we would like to get people to the market and a low-cost platform on which to do R&D is, at least one way of helping people on that journey. If you've got potentiostats and you've got screen-printed electrodes, you do need to make these two things work together. So we do have connectors. So most of our screen-printed electrodes that I discuss are 7 millimeters wide, and a lot of on our potentiostats, the banana plug to 2 millimeters. So we do need connectors, and so the connector that people would need is this kind of 7 millimeter, 2 millimeter banana plug connector. And I have put a link here in the video. People do ask, can you give me packs of sensors? The thing about sensors is we've got so many sensors: glucose, oxygen, lactate, hydrogen peroxide, pH, chloride, potassium, calcium, alcohol, nitrate, nitric oxide, ammonium, uric acid, sodium phosphate, sulfite, redox, ketone, conductivity, NADH, purine, fructose, cortisol, skatol, androstenone, troponin, galactose, glycerol, malic acid. We've got a lot of sensors. So if people ask me, well, I want a starting pack with sensors, then I'd probably just suggest, start with glucose, get some oxygen, get some potassium, And I've put the web store links, and I've also put those links underneath the video as well. Not the final question of the day, but a question. I'm quite interested in using electrochemistry for cell counting and cell identification. Cells are very small. Obviously, they're sort of when I say cells, I mean kind of cells that are in the bloodstream of somewhere between 2 and 30 micrometers in diameter. This is a patent, and I just bring it up because this is a patent for one of the biggest diagnostic companies in the world. A lot of sort of what called point-of-care companies that have to do some sort of cell counting are using electrochemistry. And I bring it up because actually, you know, when I look at their patents, they're often using electrodes on the same kind of dimensions that the ZP electrodes are on. I bring it up because people get this idea that actually they need micrometer or tens of micrometer dimensions in order to do what's called like impedance spectroscopy and to measure cells and to understand cell density. But actually you don't. You need electrodes with sort of millimeter gaps between them and millimeter type dimensions. And I just bring up a patent from one of the largest companies in the world to kind of illustrate the point. There's probably about 37 million measurements on cell count done in the US probably daily on point-of-care devices that don't look so dissimilar to this. There's many techniques in order to do cell counting, though I do think that impedance is one of the most sensitive. At ZP, we do have a whole sort of stack of technologies that allows people to take our screen-printed electrodes and actually put these kind of capillaries on them. And then once you put a capillary on it, you're getting much closer to a medical device, because now you've kind of got a self-filling controller volume type device. And if people don't want to, if people came to us and consulted with us, we would probably give them some advice. When you're doing cell counting, we have more than one technique, and sometimes actually we can be very specific by actually functionalizing the electrodes so that we capture specific cells, you know, and then we can kind of interrogate the surface of those electrodes and essentially count how many cells were in the original sample. And I've put a link to various electrodes from ZP there. Question number 5. We did have a question that came in. Somebody was interested in not measuring glucose by interstitial fluid. Then I would also refer you to question 1, where I use BioLink, which is a continuous glucose monitoring company. But if people are interested in these kind of microneedle types, there's plenty of this kind of material in the literature these days. At ZP, if you see that link, you will find that link underneath the video. We do have a lot of experience in microneedles and for putting microneedles through the skin. And we do actually have a whole technology stack where we're able to, and this is probably relevant to the question 1 as well. We do have a technology stack where we're able to put microneedles through people's skin, connect it to phones, send the data to the cloud, and start to do the development of wearable microneedle technology. But it's not straightforward. You know, this is wearable. In vitro diagnostics is one thing. Continuous monitoring of any analyte is definitely a harder thing. In some ways, if people are having a kind of roadmap, they should think about doing IVD before they do continuous monitoring. But we are experienced in it, and I should say we have a link as well. So questions. Routes to commercialization could be 3 years. People could be expending something like 100,000 per month on this kind of work. And I have put real world examples of that. Sensors for bioreactors, we do have these sensors. People can either buy the sensors and do the engineering themselves, or they can engage Zimmer and Peacock on paid programs. Setting up new labs, I've highlighted today some potentiostats, some screen printed electrodes, and some biosensors, and I will put that underneath the video as well. Measuring hematocrits, hematocrits and things like white blood cells in neutropenia. I've obviously checked the patterns, checked what I was thinking, but yes, you know, sort of millimeter scale electrodes are perfect for this. I think they're better in some ways because actually you can manufacture this kind of stuff quite readily. And then Microneedles for interstitial fluid analysis. I think the BioLink kind of example is this kind of scary example of what it can take to do this kind of work. But I also have put a link there that ZP does have the technology stack in order to do continuous monitoring using microneedles. And I think I may have actually forgotten to put this link, which is the contact us link underneath the video. But as always, every week ZP does a technical and commercial webinar. This is that, where we try to answer questions that have come in for the week. But you can appreciate that some of these questions are quite nuanced, and that's why we do the webinar, because it's sometimes easy to get the nuances across. So hopefully, if you found a bit that's relevant to you, find that link, and hopefully that link will help you sort of narrow in and get a more detailed answer. Okay, if you've got any questions, please don't hesitate to reach out to us at ZP. Thank you very much.